Atherosclerosis is the condition when foam cells accumulate on arterial intima to form plaque on arterial wall. Foam cells formation is triggered by the under-controlled low-density lipoproteins LDL and also contains significant proportions of LDL. As the plaque grows larger in size on the arterial walls, the plaque becomes more vulnerable to rupture due to formations of necrotic core. When ruptures occur, the content inside the plaque, including collagen, is exposed to platelets in the bloodstream, trap and activated platelets. Activated platelets will start dendriting and secrete more platelets activated to recruit more platelets to the site of rupture. Eventually, platelets that got recruited to site of ruptures and got activated will dendrite and form a matrix that trap red blood cells and form a blood clot called thrombus. This can block the blood flow in the artery. The block of blood flow can eventually lead to thrombotic stroke, which is a form of ischemic stroke. So in this video, we will aim to understand more the developments of thrombotic stroke via understanding thromboxane. So thromboxane is an important molecule in thrombosis and is also a platelet activators in the same group with ATP and thrombin. The precursors of thromboxane is prostaglandin G2. The synthesis of thromboxanes from prostaglandin G2 is facilitated by thromboxane synthase, which is a 60 kilodalton protein anchored in the endoplasmic reticulums of platelet cells. The precursors of prostaglandin G2 is arachidonic acid, and the synthesis of prostaglandin G2 from arachidonic acid is facilitated by cyclooxygenase. So let's just take a quick look at arachidonic acid and where it's come from. So arachidonic acid can come from membrane phospholipids, and one of the membrane phospholipids that contain arachidonic acid is glycophospholipids. It contains arachidonic acid at its second carbons, and phospholipids A2 can cut the glycophospholipids at its SN2 positions to make arachidonic acid. Arachidonic acid prefers its hairpin conformations and is a polyunsaturated fatty acid. 25% of phospholipids in brain, liver, platelets, and neutrophil cells contain arachidonic acids, and that includes the phospholipids in plasma and the cytosol. Prostaglandin G2 is also an important molecule in this synthesis of thromboxanes because it also gives rise to other derivatives, including prostaglandin E2, D2, and I2, and of course thromboxanes, which plays an important role in thrombosis, and the other prostaglandins play an important role in vasodilation and inflammation mediation. So next, let's talk about the thromboxane receptor. The thromboxane receptor, it's called thromboxane prostaglandin receptor. It has its N-terminals extracellular and C-terminals intracellular. It has 7 transmembrane domains, and it's a G coupling receptor. The protein is known to have two isoforms, which is the alphas and the beta isoforms, and the beta isoform is known to have more extended C-terminal. It has been discovered that the proteins have 4 important residues between the 4th and the 5th transmembrane domain that plays important roles in agonists and antagonists ligand binding, which is the aspartate 193, the phenylalanine 184, the threonines 186, and the serine 191. As mentioned, TPR or thromboxane prostaglandin receptor is a G protein coupling receptors that could coupling with the GQ families and the G12-13 family. When coupled with GQ family proteins, TPR or thromboxane prostaglandin receptor will activate PLC beta, which is phospholipase C beta, and thus will cleave phosphatidylinositol 4,5-phosphate (PIP2) into inositol 4,5-triphosphate and deacylglycerol. Inositol 4,5-triphosphates will act as a messenger to release calcium and increase intercellular calcium, and deacylglycerols will activate protein kinase C, and both intracellular calcium increase and activation of protein kinase C assists platelet aggregation. When coupled with the G12-13 family protein, TPR will activate ROGEF, which is rho-guanine nucleotide exchange factor, and this will lead to actin cytoskeleton remodeling, cytokinesis, contractions, and all of this will lead to platelet shape change. From our current knowledge of thromboxane synthesis and how thromboxane prostaglandin receptor works to activate platelets, and how thrombotic stroke arises from atherosclerosis, it is possible that we can develop a drug to target thromboxane prostaglandin receptor to lower the risk of ischemic stroke in atherosclerosis patients.